till now you should know what is pharmacokinetics, what are pharmacodynamics. So what do we mean by pharmacodynamics? Anyone? It's the action of drug on the body, right? So what does it include? It includes all the biochemical and the physiological reactions that are exerted by the drug on the body. That include the mechanism by which the drug is conducting all the reactions, that the mechanism of action of drug, and it includes also the effect of some other drug on the drug action, that is drug-drug interactions. Pharmacodynamics. Pharmacodynamics is the study of biochemical and physiological effects of drug and their mode of action. It deals with the relationship between the plasma concentration of the drug and its response as well as the duration of action. Effect of the drugs are quantitative. The type of response produced by, now there are two terms. One is effect and one is the action. The type of response produced by the drug is called effect. But how and where the effect is produced is called its action. Modification of the action of one drug by the another drug, called the drug-drug interactions. It is also an aspect of pharmacodynamics. Now, the principles of drug action. It can stimulate the effector site, it can inhibit or depress, it can irritate, it can be used as a replacement or for the cytotoxic action. Stimulation, it refers to the selective enhancement of the level of activity. For example, if a drug is acting on an enzyme, it can stimulate or induce that enzyme so that the reaction can be exaggerated or enhanced. Depression, the opposite of stimulation, that is, it means selective diminution of activity of the specialized cells. Irritation, that means it's a non-selective, often noxious effect and is particularly applied to the less specialized cells. They are generally not used for medicinal purposes except for some topical applications. Replacement, like in patient of hypothyroidism, where the thyroid synthesis is not occurring, we give the thyroid hormones from outside as a drug, that is a replacement therapy. In a patient in whom the sex steroids are not synthesized, they are given exogenously as a replacement drug. So, this refers to the use of natural metabolized hormones or their congeners in deficiency state that was normally being produced in the body, not being synthesized now, and that has been given exogenously as a replacement. Cytotoxic action, the drugs which are used to kill invading parasites or kill the cancer cells, attenuating them without significantly altering the host, is utilized for cure or palliation of infection or the cancer cells. Now, where does the drug act? The site of action of drug, it can be extracellular, like use of antacids to neutralize the gastric acidity, like chelating agents. These are the metal scavengers. These are chelating agents, they complex with the heavy metals. Magnesium sulfate acting as a purgative. These are the examples of drug acting extracellularly. Next, drug can act at the cellular level, like acetylcholine. Acetylcholine, they act on muscarinic and nicotinic receptors. So, action of acetylcholine on nicotinic receptor at the motor end plate. Then, cardiac glycosides. These are the drugs that increase the contractility of the heart. So, where they are acting at the membrane bound ATPase enzyme, sodium potassium ATPase. Effect of sympathomimetics on the heart muscle and the blood vessels. The third site of action is intracellular, like trimethoprim or sulfur drugs. They act by interfering with the synthesis of folic acid, or they incorporate 5-fluorouracil into the mRNA. Mechanism of drug action: only a handful of drugs, they act by their physical or chemical properties, like we discussed earlier, antacid. So bulk laxatives, by physical mass, they are acting as a laxative. Activated charcoal, they cause absorption of many basically poisonous substance. Antacids, neutralization of the gastric acidity. Mannitol and magnesium sulfate, they have got osmotic activity. Mannitol is used in patients of cerebral edema. And chelating agents like EDTA, dimercaprol, cholestyramine, they bind with the bile acids, and cholesterol in the gut. Mesna is used to prevent basically bladder toxicity of the anti-cancer drug cyclophosphamide. But these were only for a few drugs. Majority of the drugs, they target a discrete biomolecule. They produce effect by interacting with a discrete biomolecule, which usually are proteins. Now, functional proteins that are targets of drug action can be grouped into 4 categories. These can be enzymes, can be ion channels, can be transporters, and receptors, fourth one. However, a fewer drugs do act on other proteins, like for example anti-cancer drug colchicine, vinca alkaloids, they act on the structural protein tubulin. Now, the diagram is not so clear. These are the 4 different targets of drug action. The topmost one is the enzyme. You see, there the substrate fit in the enzyme to catalyze the reaction, and basically this inhibitor, it binds with the enzyme and inhibit the substrate to bind to the drug. Now these are ion channel, these are bounded by the protein. Now what happens? This blocker inhibit the ion channel. If the blocker is not there, the ion channel functions. It can be in case of sodium channel, potassium channel, or calcium channel. This is a transporter. It helps in the transport of the substrate from extracellular to intracellular phase, or vice versa. The drug inhibit or modulate the action of the transporter, and these are receptor. Coming to the first topic, enzymes. Almost all biological reactions are carried out by the catalytic influence of the enzyme, and these are one of the most important target for drug action. Drugs can either increase or decrease the rate of reaction. However, in physiological systems, enzyme activities are often optimally set. Thus, stimulation of enzyme by drugs are truly unusual. Mane, definitely in the physiological, in the human body, the physiological functions or the reactions are set at an optimal rate, and it's not possible further to increase the rate of the drug except in certain cases. Enzyme stimulation is relevant to some natural metabolites like decarboxylase enzyme. They use pyridoxine as a cofactor, which is an endogenous substrate vitamin. Now, if the pyridoxine is given exogenously, the rate of enzyme activity can be stimulated. Several enzymes are stimulated through receptors or second messengers like adrenaline. They stimulate hepatic glycogen phosphorylase. Now, in many of the cases, what is basically done is enzyme induction. That is, the synthesis of enzyme protein is increased. So, apparent increase in enzyme activity can also occur by enzyme induction. That is, synthesis of more enzyme proteins. We already discussed macrosomal enzyme induction in case of metabolism or biotransformation of drugs. This cannot be called stimulation because Km does not change. Km basically is the rate constant. Basically, it's the concentration of the enzyme, concentration of the substrate required for the half maximal velocity is called the Km. Now, এখানে কিন্তু Km টা চেঞ্জ করছে না because the enzyme synthesis, enzyme protein synthesis are increased, but it is not altering the reaction itself. Examples: anticonvulsants like phenobarbital, phenytoin, carbamazepine, rifampin, glucocorticoids, they are inducing CYP3A enzymes. Phenobarbital, rifampin, they also induce CYP2D6 and CYP2C89. Isoniazid and chronic alcohol consumption, they are inducing CYP2E1. Now, some chemicals like heavy metal salts, strong acids or alkalis, they denature the enzyme. They are non-specific, they non-specifically inhibit all the enzymes. They have got limited medicinal value. However, selective inhibition of a particular enzyme is a common mode of drug action. Such enzyme inhibition can be competitive, non-competitive. Competitive enzyme inhibition can be equilibrium type, can be non-equilibrium type. So, প্রথমে competitive equilibrium type of enzyme inhibition এটা দেয়া আছে। Now, what are the criteria? Number one, the structure of the substrate and the drug should be similar. Okay. Number two, the catalytic binding site of the enzyme should be same for the drug and the substrate. এই দুটো জিনিস হতে হবে, না হলে competitive enzyme inhibition হবে না। What happens? The drug being structurally similar to the substrate compete with the substrate to bind to the catalytic site of the enzyme. যে এনজাইম রয়েছে, যে এনজাইমে যে সাইটে সাবস্ট্রেটটা বাইন্ড করে, যদি সেই সাইটে ড্রাগটা এসে বাইন্ড করে তাহলে সাবস্ট্রেটটা বাইন্ড করতে পারবে না। It will be competing with the substrate to bind to the catalytic site. So for that the structure should be similar, otherwise they cannot bind. So the structurally similar drug compete with the normal substrate for the catalytic binding site of the enzyme, so that if the drug bind to the enzyme, the product is not formed or a non-functional product is formed and a new equilibrium sets in. Such inhibition increase the Km, but the Vmax remains unchanged. কেন? যদি আমি সাবস্ট্রেট কনসেন্ট্রেশনটা বাড়িয়ে যাই তাহলে কি হবে? As the concentration of the substrate is increased indefinitely, that will displace all the drug, so the maximal enzyme activity, that is Vmax, remains the same. But what about the Km? Km is the concentration of the substrate required for half maximal velocity. As the competitive inhibitor is already there, more amount of substrate is required, so the Km will increase. Such inhibition increase the Km, but the Vmax remain unchanged. That is, a higher concentration of substrate is required to achieve half maximal reaction velocity. But if the substrate concentration is sufficiently increased, it can displace the inhibitor and same maximal velocity can be attained. বোঝা গেছে? Now, these are some of the examples of competitive inhibitor like cholinesterase. They break down acetylcholine, and the competitive inhibitor is physostigmine, neostigmine. For MAOA inhibitor enzyme, moclobemide, dopa decarboxylase, carbidopa, benzaldehyde are there. Xanthine oxidase, allopurinol. These are some of the examples that can be asked in the viva, that can be there in the exams also. Competitive non-equilibrium type of enzyme inhibition. आगे criteria কি ছিল? The structure of substrate and the drug should be similar and they should bind to the same site of the enzyme. Now, this also the structure should be similar, they should bind at the same site, but the binding of the drug to the enzyme, it is either covalent bond or the bonding is very tight, so that the drug does not displace from the enzyme quickly. So, a non-equilibrium type of enzyme inhibition can also occur with drugs which react with the same catalytic site, but either form strong covalent bond or have such high affinity for the enzyme that normal substrate is not able to displace the drug. In this situation, Km is increased like before, but the Vmax is reduced, because now if the concentration of the substrate is increased, that will not be able to displace the drug. So, the maximal enzyme velocity will be reduced. Examples: organophosphates. Organophosphates, they react covalently with the stereotic site of the enzyme, cholinesterase. Methotrexate, they have got 50,000 times higher affinity for dihydrofolate reductase enzyme than the normal substrate dry hydrofolic acid. Now, coming to the third type, that is non-competitive enzyme inhibition. Here, the structure of the drug and the substrate can be different, and they bind to the site of the enzyme other than the substrate binding site. So, the inhibitor reacts with an adjacent site, not the same site, and not with the catalytic site, but alters the enzyme. They induce conformational change in the enzyme, so that the reaction of the substrate with the enzyme does not occur. So, and alters the enzyme in such a way that it loses its catalytic property. Thus, here Km is unchanged, but Vmax is reduced. Examples: acetazolamide is a drug, it non-competitively inhibit carbonic anhydrase. Aspirin cyclooxygenase, disulfiram, aldehyde dehydrogenase. Omeprazole is proton pump inhibitor used in peptic ulcer patient. H plus K plus ATPase enzyme. Digoxin, that is a cardiac glycoside, they inhibit sodium potassium ATPase. This is a graph. On the x-axis there is substrate concentration and the enzyme reaction velocity is on the y-axis. That is showing in the unit of Vmax, and substrate concentration is shown as Km. Now, this is a normal graph. In enzyme induction, Km is remaining unchanged, and the maximal enzyme velocity is increased. Vmax is increased. In the enzyme stimulation, Km decreases, but the maximal Vmax remains unchanged. And this for the competitive inhibition and non-competitive inhibition. Coming to the next topic, ion channels. Now, proteins which act as ion selective channels participate in transmembrane signaling. There are many ion channels that are located in the cell membrane, and that allows the passage of different ions across the membrane. So, we participate in transmembrane signaling and they regulate intracellular ionic composition. This makes them a common target for drug action. Now, drugs can affect ion channels, some of which actually are receptors, because they are operated by specific signal molecules, either directly called ligand-gated channels, for example, nicotine receptors, or through G proteins called G protein regulated channels. Either ligands are called basically the molecules that bind to the respective site, for example in this case ion channel, to regulate the opening or the closing of the ion channels. So, they can be ligand gated channels or G protein regulated channels. G protein সম্বন্ধে আমরা একটু পরে আসবো আমরা in details. Drugs can also act on voltage gated channels or voltage operated channels, like there are many voltage gated calcium channels, stress sensitive channels by directly binding to this channel or affecting ion movement. In addition, certain drugs can modulate opening and closing of channels. Examples like quinidine, they blocks myocardial sodium channels. They are used for anti-arrhythmic purpose. Dofetilide, amiodarone, they are blocking myocardial delayed rectifier potassium channels. Nifedipine, L-type voltage gated calcium channels. Nicotindin, they opens up ATP sensitive potassium channels. Sulfonylurea, pancreatic ATP sensitive potassium channels. Amiloride, they act on the renal epithelial sodium channels, and others are there. Now coming to the third topic, transporters. Now some basically chemicals, some materials, they are transported across the membrane by the help of the transporter. এখানে দেখো, the substrate that is binding at the extracellular phase of the transporter, after binding there is conformational change in the transporter and the substrate is released intracellularly. That is the role of the transporter. Now several substrates are translocated across the membrane by binding to specific transporters carriers, which either facilitate diffusion in the direction of concentration gradient, মানে যেখানে কনসেন্ট্রেশনটা কম আছে বেশি আছে সেখান দিয়ে লোয়ার কনসেন্ট্রেশন নিয়ে যাবে, তাতে কোনো এনার্জি রিকোয়ারমেন্ট লাগবে না, or pump the metabolite or ion against the concentration gradient using energy. Many drugs produce their action by directly interacting with the solute carrier class of transporter proteins. Examples are there like desipramine, cocaine. They block neuronal reuptake of the norepinephrine by directly interacting with norepinephrine transporter. SSRI, selective serotonin reuptake inhibitors. They inhibit neuronal reuptake of serotonin by interacting with serotonin transporters. Like they are dopamine transporters are affected by amphetamine, reserpine, they are affecting vesicular monoamine transporter 2 and others are there. Coming to the next topic, receptors. Receptors are defined as macromolecule or the binding site located on the surface or inside the effector cell that serves to recognize the signal, molecule, drug and initiate the response to it, but itself has no other function. Receptor একটা ম্যাক্রোমলিকিউল থাকে, সেখানে তার জাস্ট ফাংশন রয়েছে কি drug or the substrate will bind and it will basically affect the effector site so that the reaction can occur. It does not itself cause the reaction. The largest number of drugs do not bind directly to the effectors like ion channels or the transporters. What does they cause? They act through specific regulatory macromolecules. These regulatory macromolecules or the sites are called receptors. Now, drug receptor interaction. It can act as an agonist, it can act as an antagonist, it can act as a partial agonist. So, agonist. Basically, here the drug will bind to the receptor and will induce the physiological reaction. So, an agent will activate a receptor to produce an effect similar to that of physiological signal molecule. That is an agonist. যে রিঅ্যাকশনটা বডিতে হচ্ছিল, নর্মালি ফাংশন হচ্ছিল, ওটাকে বাড়িয়ে দেবে। Inverse agonist, opposite to the agonist. It will carry out the reaction in the opposite direction. So, an agent which activates a receptor to produce an effect in the opposite direction to that of the agonist. Very few are there. Antagonist, an agent which prevents the action of agonist on a receptor or the subsequent response, but does not have any effect on its own. So antagonist or inverse agonist মধ্যে পার্থক্য কি আছে? Here basically the drug is binding to the receptor in antagonist and is inhibiting the action of the agonist or the substrate, but it is not carrying the action in the opposite direction. In case of inverse agonist, the drug is binding to the receptor and carrying out the reaction in the opposite direction. This is the difference. So it inverse agonist produce an effect in the opposite direction, while the antagonist, they prevent the action of agonist, but they does not carry out the reaction in the opposite direction. They does not have any effect of its own. Partial agonist, as the name suggests, they will carry out the reaction in the same direction, but submaximal response, not full. An agent which activates a receptor to produce a submaximal effect, but antagonize the action of a full agonist. Ligand. Ligand is a Latin word that means to bind, ligare. Any molecule which attaches selectively to a particular receptor or site. It can be drug, it can be substrate. The term only indicates affinity or ability to bind without regard to the functional change. Agonist and antagonist are both ligands of the same receptor. Now, the two theories are there, how the drug interact with the receptor. Number one is receptor occupation theory and the two receptor state theory. So, receptor occupation theory, that was propounded by Clark in 1937. He propounded a theory of drug action based on the occupation of receptors by specific drugs, and the pace of a cellular reaction can be altered by the interaction of these receptors with drugs, which in fact are small molecular ligands. What does he perceive basically? He saw that interaction between two molecular species, that is the drug D and the receptor R, is governed by the law of mass action, and the effect E to be a direct function of drug receptor complex. This is a drug D, it's a R receptor, and they combine to form drug receptor complex DR, and that is carrying out the effect. Subsequently, it was realized that thus the interaction between the drug and the receptor is not sufficient to produce a response. There should be a conformational change. The agonist must also be able to activate or induce a conformational change in the receptor so that the effect can occur. Now, there are two terms, affinity and intrinsic activity. Affinity and intrinsic activity. What do we mean by affinity? Is the capacity or the ability to bind to the receptor is defined as affinity. And what is intrinsic activity? After binding to the receptor, the ability to carry out the reaction is called intrinsic activity. The capacity to induce a functional change in the receptor is designated as IA or efficacy. Affinity and intrinsic activity are two different things. Affinity of a drug for a receptor determines its concentration around the receptor required to form a certain number of drug receptor associations. Now, intrinsic activity of a drug is a measure of its ability to induce a functional change in the receptor. This intrinsic activity is shown numerically from 0 to 1. 0 বলে basically no activity and 1 মানে maximum. Comparative antagonists, they occupy the receptor but do not activate it. So they have got affinity but intrinsic activity of 0. While certain drugs are partial agonists which occupy and submaximally activate the receptor. And all or none law is not a must for the receptor. Now coming to agonist, they have got affinity and intrinsic activity of 1. Coming to competitive antagonist, they have got affinity but intrinsic activity 0. Examples are propranolol beta receptor, atropine muscarinic receptor, chlorpheniramine histamine receptor, naloxone opioid receptors. Partial agonist, they have got affinity but intrinsic activity lies in between 0 and 1. So they have got submaximal response. Examples, dichloroisoproterenol on beta adrenergic receptor, buspirone, serotonin receptor 5-HT1A. Inverse agonist. Now here the intrinsic activity becomes minus. They have got affinity and intrinsic activity lies in between 0 and -1 because they are carrying out the reaction in the opposite direction. Inverse agonism is manifest only in case of some receptors which show certain degree of constitutional activation. মানে generally আমরা কি জানলাম কি একটা reaction হতে গেলে the substrate should bind to the respective site, but physiologically in the body certain reactions are carried out without the presence of agonist even. Only few are there and those receptors basically are showing constitutive activation as they are carrying out reaction in the absence of agonist also. Like benzodiazepine receptors, they show constitutional activity. So that is they are partially active even in the basal state, complete absence of any agonist. Only this type of receptors can have inverse agonist. Like DMCM is the inverse agonist of benzodiazepine receptors. Now the lower ceiling response to a partial agonist is not because of lower affinity. In fact, some partial agonists can have higher affinity and dose to dose more potent than full agonist. Now, it has also been demonstrated that full agonists can produce maximal response even while occupying less than 1% of the available receptors. And there is a concept called spare receptors. Spare receptors মানে normally যেগুলো receptor কাজ করে বডিতে তার থেকেও অনেকগুলো receptor dormant stage থাকে, like beta 2 receptors, they have got spare receptors. On the other hand, for certain drugs the total number of receptors may limit the maximal response. Now coming to the next theory, that is the two-state receptor model. Now what does it show? Basically receptors occurs in two conformational form. One is RA and one is RI. RA is the activated form of the receptor and RI is the inactivated form. Normally in the body, what does the theory say? They occur in the equilibrium state. Now when an agonist bind to the receptor, they preferentially bind to the RA form. As a result, by binding to the activated receptor form, the equilibrium is set towards the activated receptor site. When the antagonist B, they bind equally to RA and RI form. So equilibrium is unchanged and the reaction does not occur. Partial agonist C, they preferentially bind to the RA form more, but they are also binding to RI form, but more to the RA form. So partial reaction occurs. And inverse agonist, they are preferentially binding to RI form, so that the equilibrium sets in the opposite direction. Coming to the nature of receptors. Receptors are regulatory macromolecules, mostly proteins, though nucleic acids can also act as receptors, like steroid receptors, steroid hormones, they act through nuclear receptors. Hundreds of receptor proteins have been isolated, purified, cloned, and their primary amino acid sequence has been elucidated. Molecular cloning has also helped in obtaining the receptor protein in larger quantity so that its structure properties can be more studied and helpful in some classifying of the receptor. Cell surface receptors with their coupling and effector proteins are considered to be floating in the sea of membrane lipids. আমরা জানি সেল মেমব্রেনটা একটা বেসিক্যালি বাই লেয়ার মেমব্রেনে থাকে, বাই লেয়ার স্টেটে থাকে, তার মধ্যে ভিতরটা হাইড্রোফোবিক আর লাইপোফিলিক রয়েছে, আর বাইরেরটা বিকজ অফ প্রোটিন দে আর মোর হাইড্রোফিলিক. Now what is seen, the cell surface receptors with their coupling and effector proteins, they are seen to be floating in this cell membrane, and the folding, orientation and topographic system is determined by interaction between the hydrophilic and lipophilic domains. Non-polar amino acid chain tend to bury within the membrane because of lipophilicity, while the polar group tend to come out in the aqueous medium. বাইরে চলে আসে polar groupটা. In such a delicately balanced system, it is not difficult to visualize that a small molecular ligand binding to one side of receptor molecule may be capable of tripling the response. There are 5 major families of receptors have been found out till now, with a well-defined common structural motif, while the individual receptor differ in their detailed amino acid sequence. Majority of the receptors, they are heteropolymeric, that is, they are made up of several non-identical units, and agonist binding site have been shown to bring about changes in their quaternary structure or relative alignment of subunits. Receptor drugটা বাইন্ড করলে যে অ্যালাইনমেন্ট রয়েছে হেটেরোপলিমেরিক স্ট্রাকচারে এটা চেঞ্জ হয়ে যায়. For example, on activation of subunits of nicotinic receptor, the protein structure they separate out and the ion channel gets opened up. Many clinically used drugs, they act upon physiological receptors. We know adrenaline. Adrenaline is a catecholamine that is normally present in the body that act on the basically adrenaline receptors. Now, there are some truly drug receptors, true drug receptors, mane that are not acted upon by any of the endogenous ligands that just bind to the drugs given. These, for example, benzodiazepine receptors are there, sulfonylurea receptor are there. There is also called orphan receptor. Orphan receptors are the receptors present in the body that does not bind to any endogenous ligand, that does not bind to any of the drug available till now. These are called orphan receptors. These may prove to be the targets for novel drugs. যেগুলো ড্রাগটা ডেভেলপ হচ্ছে তাদের জন্য টার্গেটটা প্রুভ হতে পারে. They may prove to be targets for novel drugs yet to be developed. Now, receptor subtypes. The delineation of the multiple types and subtypes of receptors for signal molecules may play an important role in development of a number of targeted and more selective drugs. For example, boli acetylcholine receptors, they are of 2 types, it has been seen, muscarinic receptor and nicotinic receptors, and their actions are different in different tissues. A concept strengthened by the finding that muscarinic receptors, আমরা কিভাবে বুঝলাম কি দুটো রিসেপ্টর থাকে, because muscarinic receptors they are blocked by atropine and nicotinic receptors they are blocked, actions they are blocked by nicotine or curare. Coming to the adrenergic receptor, Alquist was a scientist in 1948. He shown that adrenergic receptors can be of alpha and beta type. That was again classified into alpha 1, alpha 2, beta 1, beta 2, beta 3. এগুলো সব বেসিক্যালি কোলিনার্জিক ড্রাগসের উপর পড়বে সব। Muscarinic receptors can be of 5 types, M1 to M5. Nicotinic receptors can be of 2 types, NM receptors present in motor end plate and NN receptors present in the skeletal muscles. Now, এই রিসেপ্টর ক্লাসিফিকেশনটা কিন্তু বিভিন্ন মেথডে করা হয়, লাইক টিস্যু ডিস্ট্রিবিউশন। Beta 1 receptor is present in the heart, beta 2 receptor is present in the blood vessels. That can be based on the amino acid sequence present or the molecular colony. Now, তো বিভিন্ন রিসেপ্টর টাইপস হতে পারে, তো তার জন্য একই রিসেপ্টর টাইপসটা থাকে। The receptor classification is decided on a continuous basis by an expert group of International Union of Pharmacological Sciences. Now coming to the action effect sequence. Drug action is there, drug effect. These are two different things. Drug action mane is the initial combination of drug with the receptor resulting in the conformational change. That is drug action. And what the drug effect? After the conformational change has occurred, the sequence of events that follows, so that the variety of physiological reactions is occurring, that is drug effect, is the ultimate change in the biological function brought about as a consequence of drug action. Receptor serves two essential functions. Number one is ligand binding, recognition of the specific ligand, and number two is transduction of the signal into a response. These are the two functions of a receptor. Accordingly, the receptor has got a ligand binding domain যেখানে ligand bind করবে, and it has got a effector domain যেটা ligandটা bind করার পর effector site কে activate করবে. These domains have now actually been identified in some receptors. It is an example of a receptor having a pentameric structure, 2 alpha, beta, gamma, delta. The ligand binding domain is located in between the alpha and beta domain, alpha and beta helices, in the extracellular phase. And basically, this is the intracellular phase, this is the extracellular phase. And after binding, what is happening? The ion channel gets opened up. Transducer mechanism. After the conformational changes occurred, then the series of events occur. This is called transducer mechanism. Considerable progress has been made in understanding the transducer mechanisms, which in most instances have been found to be highly complex multi-step process that provide for the amplification of the signal as well as integration of concurrently received extra and intracellular signals. Only a handful of transducer pathways are shared by a large number of receptors. The transducer mechanisms can be grouped into 5 major categories. আমরা এক এক করে ডিসকাস করব with each type of receptors. So what are the types of receptors? Number 1, G protein coupled receptor. Number 2 is ion channel receptors, when the receptor is linked to an ion channel. Number 3 is transmembrane enzyme linked receptor. Number 4 is transmembrane JAK-STAT binding protein or receptor, and number 5 is receptors regulating gene expression, that include nuclear receptor. So these are the 5 superfamily of receptors. Now today we will discuss only on G protein coupled receptors, and rest of the class will be taken up tomorrow. So G protein coupled receptors. G protein coupled receptors, these are a large family of cell membrane receptors which are linked to the effector through one or more GTP activated protein called G protein. এখানে effector site এ G protein থাকে, that is GTP activated protein, so called G protein, for response effectuation. All such receptors have a common pattern of structural organization. একটা common pattern থাকে, কি থাকে? আসছে। The molecule, that is a G protein coupled receptor, has got 7 alpha helical membrane spanning hydrophobic amino acid segment with 3 extracellular and 3 intracellular loops. আমরা দেখাবো ফটোটা। The agonist binding site is located somewhere between the helices on the extracellular loops or phase, while another recognition site is formed by the cytosolic segment that binds the coupling G protein. So, this is a picture of a G protein coupled receptors. This is a 7 transmembrane spanning segment, 3 extracellular loops are there and the 3 intracellular loops. The ligand binding site is located in between the helices in the extracellular phase, and the effector site binding is located intracellularly. This is the G protein binding site. Here the G protein remains bound. The G protein is সম্বন্ধে পড়ি, float in the membrane. They are basically tetrameric. They are formed by alpha, beta and gamma subunit. So, they float in the membrane with their exposed domain lying in the cytosol that remains bound to the G protein binding site. They are heterotetrameric in composition, having alpha, beta and gamma subunits. Now, in the inactivated state, the GDP is bound to the alpha subunit of the G protein. Now, when the receptor is activated by the binding of the receptor, then what will happen? This GDP will be replaced by GTP, and when the GTP is bound to the alpha subunit gets activated and this GTP alpha subunit, they dissociate from the beta gamma dimer. Okay, so the activated alpha subunit carrying GTP, they dissociate from the other 2 units and either activates or inhibit the effector. The beta gamma dimer has also shown to activate. Beta gamma dimer is not inactive, they also activate receptor operated potassium channel, they inhibit voltage gated calcium channel, and they promote G protein coupled receptor desensitization at higher rates of activation. Now, there are 4 subtypes of G proteins. One is GS protein coupled receptor, GS, GI, GO and GQ. GS, what they do? They basically are they activate adenylyl cyclase. What does adenylyl cyclase do? They break down ATP to CAMP, cyclic AMP. What does GI do? They inhibit the same enzyme. They inhibit adenylyl cyclase, and thus they are decreasing the concentration of cyclic AMP. What does GO do? They directly opens up calcium channel. They closes. They inhibit calcium channels. And what does GQ type G protein coupled receptor do? They basically activate phospholipase C. So these are some of the examples of G protein coupled receptors. Now, G protein coupled receptors. The alpha subunit, the alpha GTP, G alpha GTP complex was formed. Now, alpha subunit, they have got GTPase activity. So, with time, they break down GTP to again GDP, and after forming, so the alpha subunit has GTPase activity, the bound GTP is hydrolyzed slowly to GDP. The alpha subunit then dissociate from the effector to rejoin its subunit beta and gamma, but not before the effector has been activated for several seconds. Mind it, the ligand remain bind to the G protein coupled receptor for milliseconds. But the effector side, the effect occurs in seconds. So the effect is amplified much longer than the lifetime of activated receptor, which is in milliseconds, and the signal has been greatly amplified. The rate of GTP hydrolysis by the alpha subunit and thus the period for which it will remain activated is regulated by another protein that is called regulator of G protein signaling. Protein spelling wrong ache. Regulator of G protein signaling, RGS. The onset of response through GPCR is in seconds. Now, effector pathway through which G protein coupled receptor function is number 1 adenylyl cyclase, phospholipase C, IP3 DAG pathway and channel regulation. These are the examples of drugs acting via adenylyl cyclase, phospholipase C, IP3 DAG pathway and channel regulation. Coming to the adenylyl cyclase CAMP pathway. So, activation of adenylyl cyclase by G protein coupled receptor. So, what will it do? It will increase the concentration of the cyclic AMP. So, results in the intracellular accumulation of second messenger, cyclic AMP, which functions through cyclic AMP dependent protein kinase A. Now, the protein kinase A phosphorylates and alters the function of many enzymes, ion channels, transporters, transcription factors and structural proteins. They manifest as increased contractility, impulse generation, relaxation. এগুলো বিভিন্ন ফাংশন রয়েছে বইতে পড়ে নিও। Now, responses opposite to the above are produced when adenylyl cyclase is inhibited through inhibitory G coupled receptor protein. পুরো opposite effect হবে। The action of CAMP is terminated by phosphodiesterase PDE4, PDE5, which hydrolyze into the 5-AMP. Now, this example of G protein coupled receptors that is bound to GS alpha beta gamma tetrameric structure. GTP is binding to it. What is happening? GS alpha gets separated and is basically opening up the calcium channel directly. It is activating adenylyl cyclase. ATP is broken down to CAMP and via protein kinase A pathway that is leading to increased cardiac contractility, faster relaxation. And this about the GI protein coupled receptor, the opposite pathway. Cyclic GMP. Cyclic GMP is not a pathway for G protein coupled receptors. In contrast to CAMP, the CGMP serves as an intracellular second messenger only in a limited number of tissues. Like vascular smooth muscle, they cause vasodilatation. Intestinal smooth muscle, they cause inhibition of salt and water reabsorption, and kidney, they cause natriuresis. There are two forms of guanylyl cyclase. One is cell membrane bound, another is cytosolic. The cell membrane bound is regulated by enzyme linked receptor. Eta pore porbe tomra. And cytosolic soluble guanylyl cyclase is located in the vascular smooth muscle and they are activated by nitric oxide, causing vasodilatation. Now, phospholipase C IP3 DAG pathway active. What does basically it mediated by G protein coupled receptor. What does they cause? They activate the phospholipase C beta, and what does it cause? Basically, it breaks down the phosphatidyl inositol biphosphate to inositol triphosphate and triacyl glycerol. IP3 being water soluble, it diffuses to the cytosol, mobilizes calcium from the endoplasmic reticulum store. While diacyl glycerol, they remain within the membrane, they activate protein kinase C and phosphorylate many intracellular proteins. Due to the increased calcium concentration, they mediate smooth muscle contraction, glandular secretion, transmitter release and others. Signaling in this pathway is terminated by the degradation of the second messenger. How? IP3 is dephosphorylated to inositol and diacyl glycerol is partly deacylated to arachidonic acid. This is an example of the GQ protein coupled receptors activating phospholipase C, PIP2 to DAG and IP3. IP3 is carrying out increased calcium extrusion from the endoplasmic reticulum store and DAG is basically activating protein kinase C. The last topic, channel regulation by G protein coupled receptor. The activated G proteins GHGIGO can also open or inhibit ionic channel specific for calcium and potassium without the need of the second messenger like cyclic AMP. The GH opens calcium channel in myocardium and skeletal muscle. GI and GO opens potassium channel in heart and smooth muscles. Direct channel regulation is mostly the function of beta gamma dimer. Physiological responses like changes in inotropy, chronotropy, transmitter release, neuronal activity and smooth muscle relaxation follow.